This is an OCR A-level biology video on meiosis, following the video on mitosis I just did. So let's just get straight into it. So there's 23 pairs of chromosomes, so 46 chromosomes in total. One from mum and one from dad for each chromosome, okay? So you have a maternal and a paternal chromosome. So in interphase, when we look at meiosis, before I only did 2 and now we've got 4, okay, 4 genes. But these are a pair, and these orange and green are also a pair. So if we take the left hand side, oops, sorry about that. If we take the left hand side to mean our blue and green one, how the hell am I doing that? So if we take, I don't know how I've changed the colour of my mouse. I'm finding all sorts of features in this program. It's crazy. So if we look at our blue one and our green one, these are both paternal. Okay, we'll just, for this, we'll call them from our fathers, so paternal chromosomes. On the right, we've got our red and orange ones. We'll call these our maternal. Our homologous pairs are the blue and red and our green and orange. And the definition of a homologous chromosome is matching chromosomes containing the same genes at the same places. They may contain different alleles for some or all of the genes. So these homologous chromosomes, so our blue and red, our orange and green, they code for the same genes, but they have alternate alleles, because one is from the dad and one is from mum, okay? So the blue, they code for the same things, but as you know about genetic variation, we've got alternate alleles for things. So a homologous chromosome is composed of one chromosome from dad and one from mum, okay? And there are pairs. And I know I've gone on about that for a long time, but I know for lots of people, including myself, that's a hard thing to get a grasp of. But I think I have now. So what we do in interphase, we replicate our DNA, don't we? And let's put on our centromeres. So each chromosome replicates, and then they form sister chromatids. So whereas one strand before was a chromosome, it's now become a sister chromatid, and together they form a chromosome. Okay, so we've still got 4 chromosomes despite the DNA doubling, and that is the reason why so many people get confused in this subject about the numbers in mitosis and meiosis. You've got to remember, when dealing with a chromosome can look like that. Oops, it can look like this. It can be a single strand, or it can be these double strands. So when we replicate DNA, we create these sort of double-stranded chromosomes, but we still call them chromosomes. So whereas we had 4 chromosomes before, we've now doubled the amount of DNA material, we've replicated them all, and we still only have 4 chromosomes. Hence, we go from 23 in mitosis to 23 in 23. Okay, you still have 23 chromosomes, and I think that's a pretty decent explanation of it, and hopefully you understand the numbers of it now. So the blue ones are 2 chromatids, 2 sister chromatids. They are genetically identical. The red, genetically identical. They're all genetically identical if they're the same colour, okay? But remember, these are a homologous pair. First of all, I just want to tell you, meiosis, a way to remember it. A way to remember mitosis was PMAT, and mitosis is PMAT times 2. Okay, so it's just PMAT happening twice, and there's slight differences in each stage of PMAT. Well, not each stage, some of the stages of PMAT. So first of all, of course, we have prophase, and we call this Prophase 1 in meiosis. This is building part of Meiosis 1. Okay, so we have, in Prophase 1, we get this crossing over. So our homologous pairs cross over. So blue and red were a homologous pair, they start crossing over. Our green and orange were a homologous pair, they cross over. And what happens when we get crossing over occur? We end up swapping bits of sections of DNA between the two chromosomes. Okay, so bits of the red chromosome will swap with bits of the blue chromosome, and you'll see that in the next stage. Of course, we're still dealing with prophase, so all the normal things in prophase happen. We see the movement of centrioles to the poles. We see the nuclear envelope break down, okay? And what I write here is homologous pairs cross over. That is the main difference in Prophase 1. Compared to Prophase, we didn't see this crossing over, we didn't get these homologous pairs, they weren't even mentioned, okay? But now they cross over, and let's see what sort of change this causes in Metaphase 1. So we've got that dead nuclear envelope, and look what's happening here. I've drawn them slightly larger this time, but we've got our 4 chromosomes with bits. So before we had just red, just blue, just red, just green, just orange, and now we've got 4. These 2 which crossed over in prophase have now swapped sections of DNA, and that causes variation. So in meiosis, we're producing sexual gametes, and we need as much variation as possible, and you should know the importance of that if you've looked at Unit 6, and I'm not going to go into it now, but variation is very important for natural selection and just evolution of species. And this is one of the main reasons why it happens. We get this crossing over, and alleles are swapped between the chromosome from the dad and the chromosome from the mum. Okay, so these homologous pairs swap genetic material around. So that's, I've represented that by drawing different colours. And of course it's metaphase, so we get them lining up in the centre again, and this spindle forms. But one thing in Metaphase 1 for meiosis, we can have independent assortment. So if you can see my arrow here, this pair, this chromosome could be where this chromosome is. So they could swap places. These 2 could swap places, okay? And they don't have to line up in that order, okay? So what we see is, I know when we're only dealing with 2 pairs of chromosomes here, that doesn't cause that much variation. But if you think about it, we have 4 possible alternatives of their positioning here, and that's just from 2 pairs of chromosomes. Okay, so these 2 could swap around, these 2 could swap around, these could swap and they stay, these could swap and they stay. That's 4 alternatives just for 2. So imagine the extent of variation we can have with 23 pairs all arranged in a random order, all switched about. The amount of variation you can get is incredible, mixed over with our crossing over. So crossing over and independent assortment create huge amounts of variation just from 23 pairs of chromosomes. Okay, so Metaphase 1, we see this independent assortment, the spindle forms, and then we see Anaphase. So in mitosis, we saw chromatids, so the sister chromatids, pulled apart. So this one here would be pulled two separate ways. You need to watch the mitosis video to understand what I'm talking about. But the homologous pairs are split up, so they're no longer paired together. They go to a separate centriole or centrosome. Probably centriole is the preferred term for our specification. But then we split them apart. So this is Anaphase One. They migrate towards the poles, just like in mitosis, but now we're seeing the whole chromosome. Then we get Telophase One, just the same. We get this nuclear envelope formation with our crossed over pairs. We see two new nuclear envelopes form, and we get cytokinesis, splitting them into two cells. And these two cells now have a huge amount of variation from the original cell. We call the whole thing Meiosis One, so this is half of it. And these are two haploid cells. Let's remember about the numbers again. Each one of these is a chromosome. And what we've done is we've split them apart. So one goes to one cell, one goes to the other cell. So now we've got half the numbers of chromosomes, so we produce two haploid cells, each with half of these, despite having two chromatids, which in mitosis, sort of the chromatid on its own at the end then becomes a chromosome again. But while they're bound together, they are still separate chromosomes, and they remain, and they become haploid cells. I don't really know how I can explain that anymore. I really hope you do understand that. The real issue here is just getting into your head that a chromosome can look like this, but a chromosome can also look like that, okay? That is a chromosome, that is a chromosome. If all the chromosomes in the cell are drawn like that, are drawn like this, okay, with half the number of chromosomes in here as we do in here, because all of those are chromosomes. In mitosis, we end up, in the final cell, we end up with this sort of chromosome in the final cell, but we see the full amount from the original cell, and we call that's a chromosome, and therefore it remains diploid. So that is the issue in this topic. It does my head in, and it's confusing me right now. The more I speak about it, the more confused I get, but in the back of my head I still understand that. That's a chromosome, that's a chromosome. I'm sorry to anyone who understands it, but I'm trying to get it into my own head as well for the exam, which I'm taking in a few weeks' time. Yeah, that's basically it. So if you don't understand that, just please watch my mitosis video. Please re-watch Meiosis One and try and get your head around how that works. So let's get away from that nonsense now, and let's look at Meiosis 2. So we've got this haploid chromosome. It goes into prophase, so our nuclear envelope breaks down. And let's remember, we produce two haploid cells, so whatever's happening in this cell is also going to be happening in this cell. So prophase, we see the nuclear envelope break down, and we start to see some spindle fibres form, which I didn't actually draw on in these diagrams. But then it goes into metaphase, so then these chromosomes line up just like in mitosis, and our spindle fibres form. The same will happen in this cell. So nuclear envelope broke down as prophase 2. Then we line up on the equatorial plate in the centre. The spindle fibres form, and that's metaphase 2. The spindle fibres attach to the centromere, and they then pull our chromosomes towards the centriole. And let's just draw that on the second one as well. Now we've got these single-stranded. These sister chromatids are pulled towards the centre. Okay, these are chromatids. Each line is a chromatid, but when they're pulled towards it in Anaphase 2, and then in Telophase, when they get in that nuclear envelope, call them chromosomes again. It remains haploid because we're dealing with this sort of chromosome, not that sort of chromosome. So this, not that, they're both chromosomes. We still have the same number, even though the size of them is halved. We still have 2 haploid cells. Meiosis 2 is pretty much, in a way, the same as mitosis. And in the end we have telophase, so the nuclear envelopes reform, and then cytokinesis, where we get the cleavage furrow and the 4 new haploid cells are produced. One final thing is, we had independent assortment in Metaphase 1. The same thing happens in Metaphase 2. They can line up in any order, any orientation that they want. There's nothing forcing them into particular orientations. So we get that independent assortment, and we get more variation. The only thing that doesn't happen again is prophase. We don't get any more crossing over. That only happens in prophase 1. Prophase 2, we do not get any more crossing over. That is meiosis. I hope I explained that well. It's something that you've got to watch a few videos on. It's one of my favourite topics to teach on here, because all I have to do is diagrams, and I don't have to do a load of text like this, which A, takes ages to write out, and B, is not that fun to watch. But these diagrams, I think, really help to put in perspective what's happening here. And anyone struggling with these two things, the point I'm going to make again and again, this here in mitosis, we call these chromosomes, but we also call these X-shaped things chromosomes as well. So when these X-shapes split into 2, we actually, and the 2 go into separate cells, we do not lose any chromosomes in effect, because all these X things here are 2 chromosomes which become sister chromatids. They're duplicates of each other. We don't actually lose any genetic material. Okay, so that is meiosis. I'm sorry I've gone on for a bit. Hopefully you understand that now. All the best. And next video I'm going to be looking at this differentiation of cells in plants, animals, tissues, and having a look at stem cells. All the best.